today, welcome to our very last lab. We're doing human senses, so I have lots of activities for you guys to do up at the front. So let's get into it. So congratulations on making it to the very last lab. You guys did it, so I hope you guys are very proud of yourself and everything. Oh, I should also say, I have like laryngitis right now, so if you can't hear me, please let me know. I'm struggling a little bit today. But also good luck on your exams and your finals, so hopefully those go well for you guys. And also don't forget about the idea surveys. This is what I use to improve myself as an instructor and as your teacher. So if you have any feedback for me, please give that to me. I would very much appreciate it. So this week in lab, we are going to be looking at senses, and senses are basically the biological systems that our body uses to gather and process the information from the outside stimuli. So essentially, sorry about that, like what outside stimuli can we take in and how do we interpret that? And so we have 5 primary senses: our sight, hearing, smell, taste, and touch. And so our sensory organs are a group of sensory cells that respond to specific types of stimuli. And so this little guy down here, this is called a homunculus. And so what these, basically what he's depicting is that this is what a human would look like if they were proportionate in the amount of sensory neurons. So that's why the hands are super big, the lips are super large, and that's because of the sensory cells located in those appendages. And so today we'll conduct experiments that look at 3 of our senses: sight, hearing, and touch. Oh, and I also brought in some cookies today, so that could be the taste aspect as well. And so, excuse me. For the first part, we will be looking at the eyes. So the light enters the eye through the cornea, which is going to be that kind of clear, roundish part at the front of the eye. The pupil, which is the cavity inside the eye, is going to allow the light to enter into the eye itself, and the iris is around the pupil, which will contract in response to light stimuli. So that's what happens with that. The lens behind it, this is what's going to refract the light into the retina, so you can basically pick up that stimulus and everything, and the retina is on the back of the eye here. The optic nerve, this is what's going to transmit that signal to the brain, and so there's actually a blind spot where the optic nerve lies, and so you guys will look at that today. And so, excuse me, sorry. And so for the visual experiments for today, we have after images, where you'll stare at a piece of paper, and then you'll put a white piece of paper in front of it, and you'll see a different color. We have superimposition, where you should be able to see a hole in your hand if you do this essentially. Just follow the packet and everything. It's pretty cool, like an optical illusion. And we have also an eye chart, which is on the door back here, and I will close that for you guys to use that. The line for that chart is right here. So you guys will just stand here and do a visual acuity test. And so for the next part, we'll be looking at ear function. So the ears, how they work is that we hear sound through sound waves. And so this is going to be vibrations in the air. And so the sound waves are going to enter into the ear canal, and they're going to hit the eardrum and then cause vibrations to occur. And that's going to basically influence the hairs inside the cochlea, which is the snail-looking thing right here. And so those vibrations is what's going to transmit that signal to your brain to tell you what you are hearing. And so we're going to do a couple activities with that. And so what we're also going to be talking about today is different types of hearing loss. So this is one of the most common forms of hearing loss for people. The first is any damage caused to the hair cells. So this could be as a result of loud speakers, someone shouting in your ear, something like that. Basically the hairs dislodge, and it causes hearing loss because it can't be replaced. And there's also conductive hearing loss. So this results from if there's something lodged in your ear, earwax buildup, other things like that, and that creates basically no conduction within the ear canal itself. And so there's also a mix between hearing loss, so you can have a mixture of sensorineural hearing loss and conductive hearing loss. And so sound travels through air, so what we're going to be able to do is basically see how well that we can locate our sound using tuning forks. And and then like go to different parts around your head to be able to see if you can hear that frequency. You can also see if you can hear the sound through your bone. Excuse me, sorry. You'll hit this and put it like on your bone right here. And you should be able to hear the sound travel through your skull. So it's pretty cool. If you ever see those headphones that don't go in your ear, but go on like the side of your ears right here, that's how those are actually working, is it's transmitting the sound through jawbone. That, and then for the last part, we'll be looking at touch. And so our skin has 3 primary mechanoreceptors that can detect temperature, pain, and also just touch itself. And so the mechanical stimulation is turned into electrical impulses, and this is what tells our brain if something is like hot, if it's painful, other things like that. And so we're going to see how sensitive that you are to touch. So here. And you'll just move these to like a certain distance, and you'll test it on like different parts of your body to see how sensitive it is to your like touch essentially. And lastly, I just want to thank you all for being in a really great class. I really enjoyed this semester, and I hope you guys had fun and everything. Make sure you fill out your manual as you work through the different procedures. And also don't forget we're dropping your lowest grade, so if you don't want to fill out today's packet, it's going to be your lowest one. That is an option. Don't forget about the idea surveys, and also have a great winter break. So I hope you guys enjoy your semester and also the wrap up and everything. With that, you guys can go ahead and get started. You guys can work as a table or in pairs, whatever you guys prefer. Oh, these are the focusing needles. These are sharp, so please be careful. Don't like poke your lap harder or anything. So I have those in here. And then if you guys would like a cookie, I have these up here as well. So you guys go ahead and get started, and I'll be right back.